Thanks very much for the invitation. And my lab has been involved in gene therapy for the last 30 years or so. And we have been primarily interested in making viral vectors to introduce genes into a wide variety of tissues, cells, and organs. And eventually, of course, other people have done clinical trials with the hope of finding ways of correcting genetic defects. Now, of course, there are limitations of those. So there was a possibility when the opportunity came to do cell therapy, and particularly in the coming of the iPS cells, we began to think maybe there are opportunities where we can begin to combine gene therapy approaches with cell therapy approaches, and perhaps that will come to be a common cause of making a better way of doing cure of the variety of genetic diseases. So by way of an introduction, I start with this slide which everybody shows. You have to show these days what you can do with these guys. So we have patient-specific iPS, that's the standard technology. And our lab is really involved in 3 different types of cells. We're interested in lung epithelium cells, the work of Amy Firth, who is one of the organizers of this meeting, the hepatocytes, and then, of course, the hematopoietic stem cell, which will be the primary subject. So by way of introduction then, in terms of lungs and hepatocytes, we have really relied primarily on how does the normal lung develop in a normal mouse or a normal human, and learned from that and use that technology to convert a iPS cell or an embryonic stem cell into a lung epithelial cells. So we use basically conversion of an iPS cell into mesodermal definitive endoderm, anterior endoderm, lung progenitor cells, and eventually we get the mature proximal airway epithelium. So basically using the methodology by which a normal lung develops. And doing that, Amy was able to then create some very nice lung epithelium cells which she recently published. And the idea is there are really 4 kinds of cells that we need to have these epithelium: the Clara cells, the goblet cells, the basal cells, and ciliated cells, and we were able to make all of them. I just show you one of them, which is my favorite, is the ciliated cell. They just look very pretty. And in fact, they are particularly interesting because a lot of ciliopathies can be now begin to done, and she has started collaborating with Chris Kinton and other people. The second area of interest, again, for us, just by way of background before I tell you about the hematopoietic cells, is the liver. We think liver is a fantastic organ for a wide variety of genetic diseases. And second, there's a lot of interesting things going on in making organoids from liver, and it's a possibility that if you can make hepatocytes in vitro, that you may be able eventually to do transplantation, because one of the problems with making many of these tissues, making cell lines, including, for example, lung, eventually will come the question how you're going to introduce back into people, what's the mechanism that you will introduce to do that. And liver offers an easier component to start thinking about it. And once again, Shivashni, who did most of this work, is also here, basically relied on the technology of asking how does the normal liver develop, and used the same technology, essentially different kinds of factors, and was really able to generate human liver cells, hepatocytes, from iPS, human iPS cells, which are quite functional. You can see they're making large amounts of the albumin after a certain period of time. And the reason we are really interested in the liver is that we have a very nice mouse model system. Because one of the things when you make all these cells in vitro, you still have to functionally test them. It's true you can show 1 or 2 properties or even 3 properties, but actually you have to show them, put them back, and see if the organ will behave like the normal organ in a normal individual. So here we were lucky because many years ago, Marcus Grompe had a mouse where they knocked out a particular gene, either femoral acetohydrolase. As a result, these livers are black and they die, so the mouse doesn't survive. But if you give them a drug called NTB, that allows the further downstream things not to happen and the animals survive. So we did a very simple experiment, we meaning in this case Dimi Bissek in our lab, and he basically showed the following, that if you take a human cadaver, hepatocytes, put them in these mice and start giving them, reducing the dose of the drug you put in, he actually reconstituted over 95% of the mouse has a human liver, to the point that he could actually infect them with hepatitis B and hepatitis C viruses, which are strictly human tropic. So now when Shivashni made her hepatocytes in vitro, she was able to take them, put them in these mice, and show they function like a normal liver cell. We are now waiting to see how long they will do it, but they are doing that. So I think that adds to to the reason of doing these things that we have actually an additional way of proving that the organs or the cells that we make have the potential to actually function like the normal one. And of course, this is an important component you just heard from other people. Basically, one nice thing to do here in the case of all these diseases is that one can now easily grow from a wild type by knocking out the gene to make a mutant, or from a mutant by adding the gene to make it a wild type, and then do differentiation and compare the two. So there's an isogenic component that what you're doing in one is actually phenocopied in the other and not completely different sets of genes behind them. So the example I give you is one of a X-linked combined immunodeficiency disease. And this is what the easiest way for most of you is to think about David the bubble boy, who could never get out of his bubble because he had not made one particular type of his cell which did not make T cells and B cells, and as a result he was always easily infectable. Well, the reason for that was, or in his case, or in this particular case, is a deficiency of IL-2R gamma. This is a chain which is necessary to make many other lymphokines. For example, it's the gamma chain, the third chain of IL-2, 4, 7, 9, 15, 21, for example, and if these lymphokines are not made by virtue of deficiency of this one particular receptor. So if you could provide this receptor back to it, this will complement that deficiency, and you should now make it a perfectly normal individual out of it, be it mouse, or eventually people have done in the case of human. So we have a profound defect in the cellular and humoral immunity. They lacked in basically NK cells in this case. So we obtained some sample from a patient, a small child, where we got a bone marrow from them essentially, and from which we were able to then eventually make iPS cells. So you have a SCID patient from which you can make iPS cells, but they're basically unable to make NK cells because they have a deficiency in this case of an IL-2 receptor. As a result, they don't make any of these. So basically, you reach this point, but then you can't do anything further. As a result, you have no B cell, no T cell, no NK cell, and therefore the child will be completely susceptible to, in this case, to all immune infections. However, people have done before, and there have been several clinical trials, where you can use a retrovirus, for example, or much more common now, a lentivirus, that you can introduce this particular gene, and now you're able to actually bypass this restriction because all these lymphokines are made, and this patient actually, number of them, almost now, there are almost 700 clinical trials using lenti and retro, and about 40 of these patients, or some of them, are over now 5 or 6, or maybe some of them 8 years, that they're perfectly normal in terms of making T cells and B cells. So the idea is that you can do it by introducing this one deficient gene into a deficient cell type, in this case the hematopoietic stem cell, and therefore you can reconstitute the entire hematopoietic system. However, as some of you do recall, at least in the case of retroviruses, where the integration was non-specified, in this case L number 2, so they were, of the 7 first that Alan Fisher did, 4 of them actually had leukemias. This has been alleviated much more in the case of lentiviral vectors, largely because they are SYN vectors, they don't have the LTRs into them, and they have, so far, hasn't been any example where you actually have any activation of an oncogene. So I think in some ways, the problem of this is relatively solved by using a different kind of vector. And even people using retroviruses are using where LTR is now removed, and they have actually also much better prognosis. So from our point of view then, we took the bone marrow cells, as I said. In this case, we got a very small vial because they have a little bit left, came from Australia to us from Alexander's lab, which were then converted. These are probably most like mesenchymal cells in this case. They're eventually converted into iPS cells, and now we ask the question, can we take these iPS cells, correct them with this genetic defect, can we actually make T cells or NK cells from them? And that's not an easy proposition because to my mind, no one has yet done NK or T cells from any of these iPS cells. Well, this is a standard again to show that these iPS cells are multifunctional, and then you add the correction. Now, of course, everybody has probably several slides here using CRISPRs and so on, but when Tushar started this experiment, he has actually a poster outside to give you more details. He used the TALENs. And the idea is very simple in this case. The TALENs are able to either introduce the gene or they can knock out the gene. And what he basically did in this case was to first have to sequence the genome, in this case, and find that the mutation was actually in an A to C residue right at the point where there's a splice junction. So basically what happened in this particular patient, that there was a mutation in the splice junction of the IL-2 gamma gene, and therefore they were unable to make the correct IL-2 gamma receptor, and therefore the defect. So he then made so you can actually see here that in the control you have a nice transcription of messenger RNA, where in the SCID they have a multiple message, almost none of them of the right size. Then what Tushar did then to make TALEN molecules out of it, and he can tell you more details, and we expected the size correction over here, and was able to correct them. This is the correction. He was able to correct them. In addition, he also introduced a number of silent mutations. It's a clever way of making new restriction sites so you can do more other manipulations with it. These cell lines that he generated, that by correcting this, have all the characteristics of the iPS cell lines. Here, if we were to compare a wild type cell from which we make iPSCs, or the mutant one, or the corrected one in this case, we were able to at least show they can all have the pluripotency genes into them. And here is the correction that he was able to show in the precursor cells. Here is the wild type, and here is the mutant, and here is the two corrected iPS cell lines. So we were able to actually correct them, and now the question is, can you go on to show that these corrected cell lines, iPS cell lines, can be differentiated into subsequent differentiated cell types, including the NK cell types. Now, over the years, many of us, many people here, have also done to show that the myeloid lineage is not an issue. You can take iPS cells from mouse or human and do pretty good with all the myeloid lineage. We can make GM cells, we can make erythroid cells, we can make CFU erythroid cells, either from the mutant cell or the corrected ones. That is not a problem. The problem has been the other component of it, the lymphoid component. Do you actually make T cells or do you make NK cells? Now, I'm not an immunologist, so I always have to look at these things to make sense out of it, and I presume many of them you are like me. It is really quite a process by which a human embryonic stem cell or a hematopoietic stem cell goes through the process of going to become an NK cell, mature NK cell. It's a series of changes that occur. And don't worry about looking at it. The only point mainly to mention is that the mutant SCID cells, the line that we made, the iPS cell, they come up to this part where there are pre-NK cells. They do not make the NK cells. But if we now able to do some manipulation that I'll show you in a moment, we can actually go up to this point now, where after we do the correction. So before correction, everything is stuck here, but after correction, we can actually have the ability to now make NK cells, which is one of the components of the deficiency of these disease in these patients, which can then be presumably corrected. Now, this is the work really done in collaboration with Zari Zak and his colleagues at UCLA, who are really the experts in this. And we were really very, very fortunate that they took up on to join us and do this project. So here we start these cells. These are our iPS cells here in this case. Here is the human embryonic stem cells. And again, these are all markers for the NK cells, some for mature, some for less mature, some for toxic, some for the negative receptors. But these are really the indicators of that. And by day 18, actually, the corrected one and the mutant actually really don't show you much compared to the human embryonic stem cell. So that's a disappointment. Most people would have stopped it, but Jerry went on and realized that if you now go to day 28, he began to see corrections. So you can now have the more mature markers on CD94, CD56, or even CD16 here. Now you can begin to see that you are now making, in this case, about 50%, in this case, about 36%, and the mutant actually has no NK cells. So this is actually a very nice proof that by introducing the correct gene in these iPS cells, we are able to convert them by using this technology to make them NK cells, which are now beginning to be made as compared to the human embryonic stem cell, but are absent in the mutant, as you might expect. Now, if you go on further, for example, here now things get better and better. Here again, you have the markers. This is the human embryonic stem cell, which have the ability to be making pluripotent T and NK cells. Here is the corrected version, and here again, there's nothing in the case of the mutant cells. You can now go on to do those cells which are responsible for antibody, because eventually the job of NK cells is to kill other cells. And again, you can see using those markers that the corrected ones are making Not as great, but pretty good compared to human embryonic stem cell. But again, the mouse, the mutant from which we started from the patient is unable to make, again suggesting why they are incompetent to handle because they can't make the NK cells. So it seems that we have been able, in collaboration with Jerry, to do one important aspect, and that is to convert them into NK cells. We still have not made them into T cells. There are occasionally glimmers, but we have actually not seen a long-term glimmer as yet. But it's a matter of time where if us or somebody else will do it. So I just finished my talk by making some general statements. I think it is fair to say, to date, the best transplantation organs that we have to this date is still bone marrow. And everything that we know, if we can do with bone marrow, it will be tremendous. But unfortunately, no one has yet succeeded making a hematopoietic stem cell which is like the hematopoietic stem cell we can make from human or from the cord blood, which can reconstitute the entire lineage of the blood, that is to say, the complete myeloid and the lymphoid. So that's one big question in our field, and there could be, if people have interesting ideas worth talking about. The second big question is, it will be nice if we could keep these hematopoietic stem cells alive for a longer period of time, because we are doing a lot of corrections using this CRISPR technology, TALEN technology, but that requires these cells to be around for a certain period of time, and they are not. And in fact, if you want to do a better job, you need some selection, and we can't do that. So if people can find out, recently there was a paper, somebody has a small molecule which functions, and of course we have to now try that, how reproducible it is. But I think those will be two very big things. If we can do that, will be a big part of gene therapy, cell therapy, that we can combine the two together. Finally, I can't escape the fact that being somebody who comes from gene therapy has to ask the question, what is the advantage and what's the disadvantage versus cell therapy? Now, the advantage of the gene therapy, obviously, is it's not patient-specific. Imagine somebody has factor IX deficiency hemophilia. You take an AAV vector or lenti-vector, you do a single injection. You don't care what type of a patient it was. Whereas in the case of cell therapy, you're still very much dependent on making cells for individuals. So it's a big difference. If you can find a way of doing gene therapy, it certainly has a great advantage. No need to make cell or organ transplantation. If you want to make liver, iPSC, you make hepatocyte, you don't have to do it because you can go directly into the liver or a lung or a brain or different tissues, which you can't do that right now with iPS cells. It's certainly more economical. But of course, the problem is the long-term production of the deficient protein requires the vector to be integrated, and that has the potential of insertional mutagenesis. I told you that that's not as yet the question with lenti, but it's a matter of time before we find that. Other cases, we have AAV, but they can only go as long as the tissues don't divide. The month they start dividing, you will lose it. So that's a big disadvantage. And the second big disadvantage is the host immune status. For example, 70% of people in this audience have probably AAV antibodies. If you now come with a vector which makes AAV and you have antibodies, you'll have a hell of a time trying to transduce these people. And that's a serious problem with certain vectors. But again, people say we can change it, but right now that is not the case. So I think the two technologies are quite complementary to each other, that if we can find a way of putting the two together, I think it will be a tremendous success for the regenerative medicine. Finally, people who did the work. I don't do any work, I just talk about them. Amy and Tushar are the primary people who did the work. Very Jack, fundamentally essential Adri and Zoran to us. And Ian was very kind to share with us the very small amounts of samples. And finally, none of this would have been done to us without the CERM funding. So that's the glue that really make people like me working on different areas to come together and combine the two. So thank you very much to the CERM folks.